You know, we're always hearing that we're drowning in data, but more information doesn't automatically mean more clarity. This is a massive challenge, especially in biology. So today, we're going to dive into a fascinating new method that's kind of like a magnet for the truth. It helps pull the real, meaningful signal out of a huge mountain of scientific noise.
So, put yourself in the shoes of a biologist for a second. You've just run a big experiment, and you get back thousands upon thousands of data points. How in the world do you find the important stuff in all that noise? Answering that question isn't just some technical puzzle. It's literally the key to unlocking real scientific breakthroughs.
And this data flood, it's especially intense in a field called metabolomics. It's an incredibly powerful area of study that basically gives us a snapshot of everything that's happening inside a living system, right down to the molecular level.
So what is metabolomics? Well, in a nutshell, scientists measure all these tiny little molecules. They're called metabolites, in a sample of, say, blood or tissue. We're talking hundreds, sometimes even thousands of them all at once. This creates this amazingly detailed picture. But, as you can imagine, it also generates a massive, almost overwhelming amount of data.
Now, to wrangle all this complexity, scientists have a go-to tool. It's called Principal Component Analysis, or PCA for short. Think of it like this. If you're panning for gold, PCA is like the first big sieve you use. It helps cut down all the complexity and shows you the biggest, most obvious patterns in your data. But how you use that tool is where things start to get really interesting.
See, for years, a sort of common practice has sprung up around how to interpret these PCA results. It's a kind of shortcut, and while it seems pretty logical on the surface, it has this subtle but really significant flaw that can actually lead researchers down the completely wrong path.
So here's how this shortcut usually works. A scientist runs the PCA, they look at the results, and then they just pick a number. They'll say, okay, let's just look at the top 10 most important metabolites. The problem? That number, 10, is totally arbitrary. It's just chosen out of habit, not based on any actual statistical proof.
And this is the critical point, which the researchers behind this study highlighted themselves. This subjective process creates a biased picture. It's like putting on blinders and deciding to only look at a tiny, hand-picked fraction of the whole story.
And this right here is the core of the problem. What if the true effect of a new drug involves a really subtle change across, say, dozens of metabolites? Or what if a particular disease is linked to a very specific small group of only 3? If you're always just looking at 10, you're almost guaranteed to miss what's really going on.
So, this is where the new research comes in to save the day. The authors, Yamamoto and his team, proposed a brilliant way to replace all that guesswork with actual proof, bringing some much-needed statistical rigor back into the process.
Instead of just picking the top 10, their method uses a proper statistical test on every single metabolite. For each one, it asks a really simple question. Is your connection to this biological effect statistically real, or is it just random chance? And only the ones that pass the test actually get selected. No more guessing.
And this just lays it all out, plain and simple. We're moving from a totally subjective guess to an objective, evidence-based selection. Instead of a fixed, random number, we find the true number of metabolites involved, whatever that number happens to be. It's the difference between making an educated guess and building a real scientific proof.
Okay, so theory is great, right? But does this thing actually work in practice? Well, the researchers put their new method to the test with 2 real-world datasets from some experiments they'd run with mice.
First up, they looked at the effect of fasting. Pretty straightforward. They took liver samples from normal mice and then from mice that had been fasting for 12 hours. They measured a total of 282 different metabolites to see what changed.
So what did they find? 10? 20? Nope. Using their new statistical method, they found that a whopping 136 metabolites were significantly changed by fasting. Not 10, 136. This right away shows you just how limited that old approach really was. Okay, so for the second test, they looked at something a little more subtle, the effect of a diabetes drug. They compared diabetic mice who got the drug to those who didn't, and this time they measured 275 metabolites. And what happened here? Was it 136 again? Not even close. This time, the number of significant metabolites was 66. Again, nowhere near 10. And just as importantly, it's a completely different number than what they found in the first study.
When you see these two results side by side like this, it is just so incredibly clear. We have 136 key players in one situation and 66 in another. This is definitive proof that biology doesn't fit into a neat little top 10 box. The number of important factors changes every single time, and our methods have to be smart enough to reflect that reality.
And look, this wasn't just a numbers game. By identifying the correct group of metabolites, they uncovered real, meaningful biological stories. In the fasting mice, they saw that pathways related to energy use were shutting down, which makes perfect sense, right? With the drug, they detected this subtle activation of glucose pathways, confirming the drug was actually working. These are the kinds of crucial insights they might have totally missed with the old top 10 method. So, what does this all add up to? Well, it's about a lot more than just a new statistical formula. It's really about a fundamental improvement in how we do science and how we interpret incredibly complex data. I mean, when you really boil it down, these advantages are a total game changer. By letting the statistics guide us, instead of just old habits, we make sure that the data itself gets to tell the story. And that leads to more reliable discoveries and, frankly, a much more honest understanding of biology.
You know, the authors themselves couldn't have said it any better in their conclusion. To get to the real truth, you have to be rigorous. You have to use statistical tools to make sure your conclusions are built on a solid, unbiased foundation. I mean, that's the heart of the scientific method, right? It really makes you wonder, doesn't it? If this one simple shift from just habit to statistics can so dramatically change our view of the data, what other major discoveries are just sitting out there, hidden in plain sight, just waiting for us to find the right statistical key to unlock them?